Basics of Medical Biochemistry. First edition, 2022. Chapter 9. Introduction and Overview of Metabolism. By Professor Dr. Muhammad Jawad. MBBS, Tao, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College-PAF Base Faisal-Karachi-Pakistan. Ex-Professor of Biochemistry, Qassim University Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 9. Introduction and Overview of Metabolism. 9.1 Introduction. Metabolism. The word metabolism originates from the Greek to change. Metabolism is the sum of all the chemical reactions that occur in an organism. Cellular metabolism emphasizes all chemical reactions within a cell. The main purpose of metabolism is to provide energy and needed biomolecules as an intermediate product. The energy produced by metabolism is used to maintain and operate homeostasis, growth and division, secretion, contraction, propagation of action potentials and metabolic turnover, i.e., breakdown and replacement of cellular parts. Anabolism. It is the synthesis of new organic molecules and helps functions such as ciliary movement, contraction of muscles, active transport, and cell division, and builds nutrient reserves such as glycogen stored in the muscle cells and liver. Anabolism uses the energy produced by catabolism. Catabolism. It is the breakdown of organic substances. It releases energy used to furnish ADP. Initial steps of catabolism occur in the cytosol, producing smaller molecules but little ADP. These molecules enter the mitochondria where significant amounts of ADP are produced. 60% of the energy released in catabolism is produced as heat, whereas 40% of the energy is used to make ADP. Amphibolism. It is a biochemical pathway that includes both anabolic and catabolic processes. Any biochemical pathway which involves both aspects of chemical processes, i.e., constructive and destructive, is known as an amphibolic pathway. The TCA cycle is an excellent example of amphibolism. Metabolic cycles, a series of reactions in which the substrate is continuously reformed and the intermediate metabolites are continuously regenerated. Substances that enter the cycle are known as feeders of the cycle, while the molecules that leave the cycle are called products. The starting compound of the metabolic pathway is known as precursor. Cascade reactions. These are metabolic processes in which the product of one reaction activates the rate of a second reaction. This produces significant amplification of a response. Figure 9.1 Cellular metabolism. Normal metabolism. It occurs under normal circumstances as well as in adaptive situations like phases of starvation, exercise, pregnancy, and lactation. Abnormal metabolism. It may result from nutritional deficiency, enzyme deficiency, abnormal secretion of hormones, or the actions of drugs and toxins. Knowledge of normal metabolism is crucial for an understanding of anomalies underlying disease. Figure 9.2 Metabolic organization. The function of metabolic pathways. 1. Energy production during all conditions of well-fed, fasting and starvation, during rest and muscular exercise, during pregnancy and lactation. 2. Synthesis of biomolecule intermediates that are required for the cells, in other pathways. 3. Removal of toxic substances. 9.2 Products of digestion and metabolic pathways. The major absorbed end products of food digestion are monosaccharides, mainly glucose, from carbohydrates, monoacylglycerol and short-chain fatty acids, from lipids, and small peptides and amino acids, from protein. Once in the bloodstream, different cells can metabolize these nutrients. All the digestive end products are metabolized to a common product, i.e., acetyl-CoA, which is then oxidized by the TCA cycle to produce ADP by oxidative phosphorylation. The daily energy requirement of an adult healthy human being of 70 kg is about 8 to 12 mJ, 1920 to 2900 kilocalories, from metabolic fuels. However, it varies depending on the physical activity. It is supplied by carbohydrates, 40 to 60%, lipids, mainly triacylglycerol, 30 to 40%, and proteins, 10 to 15%, including alcohol. The three stages of catabolism of main nutrients are 1. Hydrolysis of complex nutrients to their constituent building blocks. For example, complex carbohydrates such as polysaccharides are degraded to simple sugars, monosaccharides, proteins to amino acids and lipids, triacylglycerols, to fatty acids and glycerol. 2. Conversion of all the products of digestion are metabolized to acetyl-CoA. 3. Oxidation of acetyl-CoA by the TCA cycle to yield ADP. Figure 9.3 Stages of catabolism. Carbohydrate reserves are found as glycogen in the liver and muscle and stored lipids as triacylglycerol in adipose tissue. When the intake of metabolic fuels is consistently greater than energy expenditure, the surplus is stored, largely as triacylglycerol in adipose tissue, leading to the development of the condition of obesity. When the intake of metabolic fuels is consistently lower than energy expenditure, the reserves of metabolic fuels are depleted gradually and lead to weakness, severe muscle and generalized wasting, and eventually death may occur. 9.3 Cellular metabolism. It is defined as the complex collection of biochemical reactions involving the synthesis of biomolecules, maintenance, or their breakdown at the cellular level. Molecules involved in these metabolic processes include basic cellular building blocks such as lipids, amino acids, carbohydrates, and nucleotides. Numerous enzymes and cofactors also participate in these metabolic reactions. Cells provide small organic molecules to their mitochondria. Mitochondria produce ADP used to perform cellular functions. The production of ADP within the mitochondria involves two pathways, the TCA cycle and the electron transport system. The main cellular metabolic reactions are influenced by nutrient uptake. Thus, it is crucial to understand the key nutrient metabolic processes occurring in the cell, including lipid, carbohydrate, amino acid, and nucleotide metabolism. Compartmentalization in the cell allows the adequate specific localization of the enzymes of metabolic pathways. The proper distribution and the sequential arrangement of enzymes in a cell compartment or organelle increase the efficiency at which chemical transformations can proceed. Figure 9.4 Nutrients used in cellular metabolism and role of mitochondria in metabolism, upward arrow, anabolic pathway, downward arrow, catabolic pathway. 9.4 Role of mitochondria in metabolism. Mitochondria are intracellular organelles that play a chief role in cellular metabolism and energetics. They are famously known as the powerhouse of the cell. It acts as an efficient engine that converts supplied nutrients into energy currency, i.e., ADP. The formation of ADP from metabolites in the TCA cycle is achieved by the transfer of electrons from these intermediates through the respiratory chain subunits found on the mitochondrial inner membrane. The biochemical gradient is established when electrons are transferred and protons are pumped into the inner membrane space. The key respiratory complexes I, III, and IV are found in the inner mitochondrial membrane and are the protein units that pump the protons from the matrix into the inner membrane space using electrochemical energy derived from the transfer of electrons. Finally, molecular oxygen acts as the electron acceptor from complex 4, and it is reduced to H2O. The electrochemical gradient is established by the imbalance of protons between the mitochondrial matrix and inner membrane space, which provides the potential energy to drive ADP synthesis from ADP. This efficient energy-generating process is termed aerobic respiration as it is oxygen-dependent. The function of mitochondria regarding energy generation is especially crucial in energy-demanding tissues and cells of the body. Main examples of such cells are neurons, cardiac cells, and beta islets of the pancreas. Reactive oxygen species, ROS, are produced in mitochondria as the byproducts of ADP generation via electron transport. These are highly reactive molecules that can cause oxidative damage to the cell organelles and other cellular structures. In normal limits at physiological states, ROS toxicity can be adequately controlled by intracellular antioxidant systems. These natural intracellular antioxidants include superoxide dismutase, SOD, glutathione, and catalase. It is now known that basal ROS levels are playing a critical role in physiological cell pathways. But pathological ROS levels have been shown to be dangerous and damaging to proteins, lipids, and DNA, which can lead to defective mitochondrial metabolism and harmful consequences for cell function and viability. 9.5 Overview of carbohydrate metabolism. Carbohydrates are composed of carbon, hydrogen, and oxygen, exist as simple monomers, monosaccharides, such as glucose, or in more complex forms, ranging from disaccharides, example sucrose, to highly complex polysaccharides, example starch. Carbohydrate metabolism begins in the mouth with an enzyme known as salivary amylase. Salivary amylase breaks down complex carbohydrates, i.e., polysaccharides, into less complex molecules. Digestion of carbohydrates continues in the small intestine, where pancreatic amylase further breaks down the partially digested polysaccharides into their simplest form, i.e., monosaccharides. Glucose is the primary monosaccharide in the human diet, providing much of the daily energy needs of an individual. Glucose is absorbed into the bloodstream in the small intestine and transported by the circulatory system to all organs, where its uptake into cells is induced via insulin. Glycolysis is the first step in the breakdown of glucose. It involves the oxidative breakdown of 1-glucose into 2-pyruvate molecules with the capture of some energy as ADP and NADH plus H+, which is used to create ADP in the electron transfer system. Glucose uptake is particularly prominent in liver and muscle cells, where metabolic enzymes convert glucose into glycogen, polysaccharide, through the process of glycogenesis. Glycogenesis serves an important energy storage function. Glycogen is composed of multiple glucose subunits, acts as an emergency fuel reserve, and is readily broken down when blood glucose levels drop. The breakdown of glycogen is known as glycogenolysis. HMP shunt is regarded as an alternate pathway of glucose oxidation. It neither produces ADP nor utilizes it. It involves the utilization of all forms of monosaccharides with the liberation of CO2 and many side products such as NADH plus H+. It provides pentose for the synthesis of nucleic acids, DNA and RNA, ADP, coenzyme A, etc. It also provides intermediates for other metabolisms of carbohydrates. Gluconeogenesis is the pathway to create glucose from non-carbohydrate sources. Gluconeogenic substrates include glycerol, lactate, propionate and glucogenic amino acids. It is an important pathway that allows the body to store needed energy for the brain in the form of glucose. Gluconeogenesis occurs in the liver and kidneys. It supplies the requirements for plasma glucose between meals. Gluconeogenesis is stimulated by diabetogenic hormones like glucagon, growth hormone, epinephrine, and cortisol. Clinical aspects of carbohydrate metabolism. Carbohydrate metabolism disorders result from the changes in enzymatic function within the carbohydrate metabolic pathway, which lead to several diseases, such as diabetes and a variety of glycogen storage diseases. Glycogen storage diseases are inherited disorders that result from mutations to key enzymes involved in the biochemical processing of glycogen. There are more than 10 types of glycogen storage diseases that are classified based on the enzyme that is affected. The organ system that is affected depends on the tissue distribution of the affected enzyme, therefore a wide array of symptoms can result. Glucose 6-phosphate dehydrogenase, G6PD, deficiency is another example of a carbohydrate metabolism disorder. G6PD deficiency leads to hemolytic anemia. It is a congenital hemolytic disease due to an inherited deficiency of G6PD causing the deficiency of NADH plus H+, which is required for healthy erythrocytes that do not have other NADH plus H+ sources. There occurs lysis of red blood cells because of a buildup of reactive oxygen species, and ultimately symptoms of hemolytic anemia appear. Figure 9.5 Overview of carbohydrate metabolism. 9.6 Overview of lipid metabolism. Lipids or fats are important energy-rich nutrients that cells use as fuel to support cell functions. In addition, lipids serve as important building blocks for key cellular structures like membranes. They also participate in many important signaling networks. Certain cells like cardiac cells are specific for the storage of lipids, which can be mobilized through catabolism to support organismal function in hard circumstances of nutritional or energetic deficiency. Main lipids, such as triglycerides and cholesterol, are obtained from the diet and have to be appropriately digested and absorbed. Cholesterol is essential for life, despite its deposition in arteries being associated with cardiovascular disease. In a healthy individual, a complex balance is maintained between the biosynthesis, utilization, and transport of cholesterol, keeping its harmful deposition to a minimum. De novo synthesis of cholesterol takes place in the liver from acetyl-CoA. Cholesterol acts as a precursor for bile salts, steroid hormones, and vitamin D. Clinical aspects of lipid metabolism. Lysosomal storage diseases are a group of disorders in which the lysosome is deficient in main lysosomal enzymes, thereby decreasing breakdown and increasing storage of lysosomal material targeted for degradation. Gaucher's disease and Niemann-Pick disease are two examples of lysosomal storage diseases. Gaucher disease results from the deficiency of the enzyme glucocerebrosidase, acid beta-glucosidase, and leads to the buildup of glucocerebroside, a sphingolipid in white blood cells and macrophages. Niemann-Pick disease is a lipid storage disorder in which pathological levels of lipids, mainly sphingomyelins, and cholesterol accumulate in the spleen, liver, lungs, bone marrow and brain. This disorder is also known as sphingolipidosis. 4 types of Niemann-Pick disease are classified based on their genetic basis and the associated symptoms. In the classic infantile type A variant, a missense mutation causes a complete deficiency of sphingomyelinase. Figure 9.6 Overview of lipid metabolism. 9.7 Overview of proteins and amino acids metabolism. Proteins are composed of amino acids linked in series to form polypeptide chains which, upon three-dimensional folding, form a mature protein. Because proteins constitute a significant portion of all cells, they are also regarded as the main nutrient that cells use for fuel and other functions. Proteins perform many functions like providing intracellular and extracellular structure, signaling, transport, acting as enzymes and providing immunity. Digestion of proteins begins in the stomach where stomach acid and the enzyme pepsin break down proteins into simpler polypeptides. These polypeptides are then further broken down into their constitutive amino acids that can either be recycled for the formation of other proteins or further broken down by the liver into alpha-keto acids that can be utilized for energy, glucose, fat, or new amino acid formation. A significant amount of ammonia is produced in different body tissues from the catabolism of amino acids by deamination and deamidation reactions. A considerable quantity of ammonia is also produced by the gut. Ammonia is absorbed into the portal circulation, taken up by the liver and is finally detoxified into urea. It is important to note that proteins are a very useful source of energy during times of starvation. This is because the breakdown of proteins leads to the creation of metabolic intermediates that can feed into the citric acid cycle. Amino acids may also be precursors of other compounds like purines, pyrimidines, neurotransmitters, and hormones such as epinephrine and thyroxine. Clinical aspects of protein metabolism. Amino acid metabolism disorders are genetic diseases that affect amino acid catabolism. Main examples of amino acid metabolism disorders are phenylketonuria, alkaptonuria and maple syrup urine disease. Phenylketonuria occurs in infants due to complete to mild deficiency of phenylalanine hydroxylase. Affected infants are unable to break down the amino acid phenylalanine into tyrosine. This leads to the buildup of phenylalanine in the blood. Phenylalanine is toxic to the brain, and if phenylketonuria is left untreated, it can cause mental retardation due to the deposition of phenylalanine and its toxic metabolites in the brain. Alkaptonuria occurs due to a congenital deficiency of homogentisic acid oxidase, which results in the accumulation of homogentisic acid and its appearance in urine. Similarly, maple syrup urine disease occurs in children who are unable to break down leucine, isoleucine, and valine, leading to the buildup of amino acid byproducts. Neurologic problems are common and this disease has a high mortality rate. There are increased levels of branched-chain amino acids in blood and urine. Urine has a characteristic odor as maple syrup or burnt sugar. Figure 9.7 Overview of protein and amino acid metabolism. Glutamic acid, GLN, glutamine, P, phosphate. 9.8 Overview of nucleic acids and nucleotides metabolism. Nucleic acids, RNA and DNA, are non-protein nitrogenous polynucleotide biopolymers found in all living cells. They are regarded as essential substances of the genes and act as the main information-carrying molecules of the cell. The cyclical breakdown and synthesis of the various types of nucleic acids is critical for energy storage and generation and is, therefore, essential for cellular homeostasis. Nucleic acids present in foods are digested in the small intestine with the help of both pancreatic enzymes and enzymes produced by the small intestine itself. Pancreatic enzymes called ribonuclease and deoxyribonuclease break down RNA and DNA into smaller nucleic acids. The nucleotides produced by this digestion are further broken down by two intestinal brush border enzymes, nucleosidase and phosphatase, into pentases, phosphates, and nitrogenous bases, which are then absorbed through the wall of the alimentary canal. The synthesis of purine and pyrimidine takes place by de novo synthesis and salvage pathways. Purine and pyrimidine bases created by the degradation of RNA or DNA and intermediate of purine and pyrimidine synthesis can be directly converted to the corresponding nucleotides by salvage pathway or removed as waste products. Clinical aspects of nucleic acids and nucleotide metabolism. Inborn errors of purine and pyrimidine metabolism result in various diseases like gout and orotic aciduria respectively. The study of nucleic acids is playing a significant role in cancer research. 9.91 Carbon metabolism. 1. Carbon metabolism encompasses a broad range of biosynthetic reactions that occur in the cytoplasm and the mitochondria which are essential for maintaining cellular homeostasis. It refers to a group of folate-dependent metabolic pathways that are essential for the anabolism of several molecules, such as amino acids and nucleotides. In 1-carbon metabolism, folic acid acts as a carrier of 1-carbon groups, facilitating the removal and transfer of these groups from donor molecules. 3 molecules can be used to move 1-carbon groups. I-tetrahydrofolate, a folate derivative that acts as a cofactor for several enzymes. 2-S-adenosylmethionine, a methyl donor. 3. Vitamin B12, a coenzyme in methylation and carbon rearrangement reactions. 1-carbon metabolism is also important for DNA and histone methylation. In the mitochondria, 1-carbon units can be made from serine, glycine, sarcosine, or dimethylglycine and exported to the cytosol as formate. 9.10 Cancer and metabolism. Cancer is defined as uncontrolled cell growth and division. Cancer cells need the building blocks and energy to make new cells much faster than healthy cells. Therefore, they rely heavily on glucose and rapidly convert it into pyruvate via glycolysis. Currently, metabolism is covering an important aspect of oncology. Research on metabolism showed that cancer cells rely on the inefficient metabolic pathway of fermentation rather than the much more energy-efficient aerobic respiration. This Warburg effect is a hallmark of many cancers that reflects a metabolic adaptation by the cancer cells that promote their growth and survival. This prevalent hypothesis is that these metabolic adaptations provide a survival advantage to cancer cells over normal cells, since a vigorous generation of cellular building blocks i.e., proteins, lipids, and nucleic acids in oxygen-deficient conditions is required to support their rapid and uncontrolled proliferation. Another aspect of research suggests that cancer cells need to use their nutrient inputs as cellular building blocks, thus, they switch from catabolic, nutrient-consuming, to anabolic pathways to support their high proliferation rates. The metabolic profile observed in cancer cells often includes increased consumption of glucose and glutamine, increased glycolysis, changes in the use of metabolic enzyme isoforms, and increased secretion of lactate. Other facts observed in cancer research include impaired uptake of glucose and amino acids, utilization of different modes of nutrient attainment, binding, and boosting glycolysis/TCA cycle intermediates for biosynthesis and NADPH production, higher demand for nitrogen, changes to metabolite-driven gene regulation and metabolic interactions with the microenvironment. Many side effects of cancer or its treatment may cause appetite loss, which leads to weight loss and changes to the immune system. Figure 9.8 Metabolism in normal and cancer cells, Warburg effect. 9.11 Regulation of metabolism. Metabolic pathways interact in a complex way to allow adequate regulation. This interaction includes the enzymatic control of each pathway, each organ's metabolic profile and hormonal control. The blend of carbohydrate, lipid, and protein being oxidized be different, depending on whether the individual is in the fed or fasting state and on the length of duration and intensity of physical work. 9.11.1 Metabolic profiles of main organs/tissues. Each organ has a metabolic relationship with the rest of the body, although each has its role in the overall metabolism of biological fuels. It is to be noted that all the major metabolic pathways of the body do not operate in every tissue at any given time. Liver and adipose tissue function as fuel storage tissues and reservoirs. Each organ has a unique metabolic profile. 1. Brain. It is simply a fuel consumer. At only 2% of the body mass, brain consumes 20% of body's resting O2. It can synthesize glycogen, but stores are very limited. Usually, neurons use only glucose as an energy source. Since the brain stores only a very small amount of glycogen, it needs a steady supply of glucose. During long fasts, it becomes able to oxidize ketone bodies. 2. Liver. It can use all fuels except ketone bodies as it is deficient in thiophorase enzyme. The maintenance of a fairly steady concentration of glucose in the blood is one of the liver's main functions. This is accomplished through gluconeogenesis and glycogen synthesis and degradation. It synthesizes ketone bodies when acetyl-CoA is plenty. It is also the site of urea synthesis. 3. Adipose tissue. Its primary function is to store and release fatty acids. Fatty acids are stored as triacylglycerols, but the synthesis of fatty acids in the body is limited. Glucagon activates a hormone-sensitive lipase, which hydrolyzes triacylglycerols yielding glycerol and fatty acids. These are then released into the bloodstream as lipoproteins. 4. Muscle. Muscles use glucose, fatty acids, ketone bodies, and amino acids as an energy source. It also contains a reserve of creatine phosphate, a compound with a high phosphate transfer potential that can phosphorylate ADP to ADP, thereby producing energy without using glucose. At rest, muscle uses 30% O2 of the body. The amount of creatine in the muscle is enough to sustain about 3 to 4 seconds of exertion. After this period, the muscle uses glycolysis, first anaerobically, as it is much faster than the citric acid cycle, and later in aerobic conditions. Blood-borne glucose from hepatic glycogen is quickly depleted during strenuous prolonged exercise. The energy needed in this situation is met by the mobilization of fatty acids and increased blood flow for O2. 5. Kidney. The main function of the kidney is to excrete urea, electrolytes, and other nitrogenous waste products. The kidney has a significant role in nitrogen metabolism. The excess nitrogen is first assimilated to glutamine by glutamine synthetase. Then, kidney glutaminase cleaves glutamine into glutamate and ammonia, which the kidney immediately excretes. In this way, nitrogen is excreted without affecting blood bicarbonate levels. In addition, the kidneys play a major role in the excretion of drugs, hormones, and xenobiotics. A hormone, erythropoietin, produced by the kidneys stimulates red blood cell production. Other hormones produced by the kidneys help to regulate blood pressure and control calcium metabolism. It can also perform gluconeogenesis to release glucose into the bloodstream. Figure 9.9 Metabolic profiles of different organs. 9.11.2 Hormone control of metabolism. Hormone control is mainly affected by the action of two hormones synthesized by the pancreas, insulin and glucagon. Insulin is released by the pancreas when blood glucose levels are high, i.e., after a meal. Insulin increases the content of facilitative glucose transporters, GLUT4, in the cell membrane, thus increasing the ability of these tissues to take up glucose. The binding of insulin to its receptor causes the activation of tyrosine kinase, which autophosphorylates its receptor. Insulin stimulates glucose uptake by the muscle, glycogen synthesis, and triacylglycerol synthesis by the adipose tissue. It inhibits gluconeogenesis and glycogen degradation. Insulin is an anabolic hormone, that increases the storage of glucose, fatty acids, and amino acids. Glucagon is secreted by the alpha cells of the islets of Langerhans when the blood glucose concentration falls. It acts through the activation of adenylate cyclase, which results in the formation of CAMP. Its effects are opposite to those of insulin. The main effect of glucagon is to maintain blood glucose levels during fasting. In the liver, glucagon stimulates glycogen degradation and the absorption of gluconeogenic amino acids. It inhibits glycogen synthesis and promotes the release of fatty acids by adipose tissue. Thyroid hormones excite diverse metabolic activities in most tissues, leading to an increase in basal metabolic rate. It increases the basal rate of oxygen consumption and heat production. There is an increase in plasma membrane Na plus K plus ATPase activity and a decrease in superoxide dismutase activity. It increases glucose absorption and metabolism. It also stimulates fat mobilization, leading to increased concentrations of fatty acids in plasma. At moderate protein diet, it has an anabolic effect, and in high concentration, it has a catabolic effect. 9.12 Metabolic Map. The organized collection of main metabolic pathways in a single figure is called a metabolic map. It includes glycolysis, TCA cycle, acetyl-CoA oxidation, disposal of the electrons released by glycolysis, and glycogen metabolism. It also incorporates HMP shunt, urea cycle, lipid and amino acid metabolism. It provides information about interconnected metabolic routes among different biomolecules. It also explains the effect of one metabolic pathway on another. Figure 9. 10 metabolic map showing important reactions of intermediary metabolism. Straight arrows indicate forward and reverse reactions that are catalyzed by the same enzyme. Curved arrows indicate forward and reverse reactions that are catalyzed by different enzymes. UDP, uridine diphosphate, P, phosphate, CoA, coenzyme A. Source, Liaquat's Illustrated Review Biochemistry 7th edition. Summary. Metabolism is the sum of all the chemical reactions that occur in an organism. Anabolism is the synthesis of new organic molecules. It uses the energy produced by catabolism. Catabolism is the breakdown of organic substances. It releases energy used to furnish ADP. Amphibolism is a biochemical pathway that includes both anabolic and catabolic processes. Normal metabolism occurs under normal circumstances as well as in adaptive situations like phases of starvation, exercise, pregnancy, and lactation. Abnormal metabolism may result from nutritional deficiency, enzyme deficiency, abnormal secretion of hormones, or the actions of drugs and toxins. Cellular metabolism is defined as the complex collection of biochemical reactions involving biomolecular synthesis, maintenance, or their breakdown at the cellular level. All the products of digestion are metabolized to acetyl-CoA, which is then oxidized by the TCA cycle to yield ADP. Mitochondria play a chief role in cellular metabolism and energetics. They act as an efficient engine that converts supplied nutrients into energy currency, i.e., ADP. Glucose is the primary monosaccharide in the human diet, providing much of the daily energy needs of an individual. Gluconeogenesis is the pathway to create glucose from non-carbohydrate sources. Gluconeogenic substrates include glycerol, lactate, propionate and glucogenic amino acids. Metabolic disorders result from changes in enzymatic function within the metabolic pathway, which lead to several diseases. In a healthy individual, a complex balance is maintained between the biosynthesis, utilization, and transport of cholesterol. A significant amount of ammonia is produced in different body tissues from the catabolism of amino acids by deamination and deamidation reactions. The ammonia is absorbed into the portal circulation, taken up by the liver and detoxified into urea. The cyclical breakdown and synthesis of the various types of nucleic acids is critical for energy storage and generation and is, therefore, essential for cellular homeostasis. 1-carbon metabolism refers to a group of folate-dependent metabolic pathways that are essential for the anabolism of several molecules. Cancer cells need the building blocks and energy to make new cells much faster than healthy cells. Metabolic pathways interact in a complex way to allow adequate regulation. Each organ has a metabolic relationship with the rest of the body, although each has its role in the overall metabolism of biological fuels. The metabolic map is the organized collection of main metabolic pathways in a single figure. References. Exercise questions. MCQ 1. Which of the following reactions takes place during the glycolysis of glucose? Dehydration, hydration, hydrolysis, oxidation, reduction. MCQ 2. Which of the following biochemical pathways occurs partly in the mitochondria and partly in the cytosol? Gluconeogenesis, glycogenesis, glycolysis, ketogenesis. TCA cycle. MCQ 3. Which of the following biochemical pathways involves the interconversion of all types of monosaccharides? Glycogenesis, glycolysis, HMP shunt, ketogenesis. TCA cycle. MCQ 4. An obese individual is brought to the ER. The patient has been on a starvation diet for 4 months and has lost 30 kilograms weight while consuming only water and vitamin pills. Which of the following is expected to be elevated in his blood? Acetate, acetoacetate, bicarbonate, glucose, lactate. MCQ 5. A patient suffering from a rare neuroendocrine tumor of insulinoma presented with complaints of weakness and tiredness due to repeated attacks of hypoglycemia. Which of the following is expected to be raised in the blood of this patient? Cholesterol, acetoacetate, bicarbonate, C-peptide, lactate. Please write answers in the comments. End of Chapter 9. Thank you.